Hello my dear students, myself Meenu Srivastav, and today we are going to discuss about the microbiological assay of amino acid. In this lecture, we will discuss the microbiological assay especially for the three particular amino acids. These are named as valine, leucine and methionine. Okay, so here as we know that the amino acid are essential growth factor for microorganism as well as for us also. So if from the medium, if we are reducing or just subtracting these growth factors, so the microbial growth will affect. So this is the principle, or we can say this is the main principle behind the microbiological assay of our amino acids. Now I have just defined this. The amino acids are the growth factor which are essential for the growth and multiplication of microorganism. If any essential amino acid, amino acid, okay, amino acid is absent in the culture media, then microorganism fail to grow. Okay, so in the absence of growth factor, the microorganisms are not able to grow. So the principle involved in the assay of amino acid is based on the determination of microbial growth obtained after the addition of required factor. Means if we are just subtracting the growth factor from our nutrient medium, then the microorganism growth will not at all take place. But if we are after some time, if we are adding our growth factor in a proper concentration, then the microorganism start to grow. So we want to determine the concentration of growth factor, that at which concentration the microorganism will grow at this much extent. So that concentration we want to determine. Here I have given the one list that the microorganism. Now come to the requirement part. Before the requirement, I want to say that in our nature we have 20 amino acids, only and only 20 amino acids we are having. But these 20 amino acids are arranged and in n number of arrangements and form the all kinds of protein, because the proteins are the polymer of amino acids. So only these 20 amino acids are arranged in n number of pattern and just form such kind of the proteins. And these proteins are the basic constituent of our cellular system as well as the microorganism cellular system also. So the microorganism also having the requirement of the amino acids in their nutrient medium. Here first we are going to cover just requirement part. First is the culture of the test organism. In the first requirement, the culture of microorganism, test microorganism which require the amino acid for growth. Here I have providing some microorganism for proper amino acid, because the particular amino acid is required for a particular microorganism. So here the Streptococcus faecalis, the amino acid is the valine and leucine. The microorganism is the E. coli, the amino acid is leucine and glutamic acid. If the microorganism is lactobacillus, the amino acid are several amino acids. And not several is not named, several means the different amino acid we use the lactobacillus. Okay, the microorganism, if Leuconostoc mesenteroides, the amino acid is methionine, histidine and leucine. Okay, now spatial nutrient medium. In the second requirement, the spatial nutrient medium means the nutrient medium including all essential nutrients except the factor under the test. Okay, so we have to take that particular medium which contain all the essential mediums except that content which want to test. Okay, so here we want to test the amino acids, so that amino acid will just subtract from that medium. Now come to the procedure part. Here prepare the nutrient medium, and for nutrient medium is the Bacto amino acid assay medium. Here is the medium, medium. Prepare nutrient medium and that named as the Bacto amino acid assay medium. So Bacto amino back to amino acid assay medium is medium which do not have the amino acid in their content basically. So here in the procedure, in another set of the now come to the another step of the procedure. Now before going to the second step of the procedure, I want to remind you that all the glassware must be sterilized, otherwise your all assay will just spoil. Now the second point, in another set of test tubes add graded concentration of standard amino acid. First you will prepare the stock preparation. For the concentration of 200 microgram per ml. The concentration is 200 microgram per ml. But you just take the test tubes and add the graded amount of the concentration, and these are the 0.1 ml, 0.2 ml, 0.3 ml, 0.4 ml and 0.5 ml. Okay, so like this you will just add 0.1, 0.2, 0.3, 0.4, 0.5 ml in 5 test tubes. Now the main thing is that you should prepare the triplicate for all test tubes. In the amino acid assay, in the assay of turbidimetric method, we prepare the duplicate. But in this amino acid assay, we prepare the triplicate. Means for 0.1 ml, just take in 3 tubes. 0.2 ml, take in 3 tubes. 0.3 ml, take in 3 tubes. 0.3 ml, 0.3 ml, 0.3 ml, 0.3 ml. 0.1 ml, 0.1 ml, 0.1 ml. Like this, you have to prepare all the test tubes in the three types. You can you say that in the triplicate manner you will prepare for the each and every preparation. So you can reduce your reduce your faults. Okay. Now similarly, take a test tube containing a sample whose strength we have to determine. Now take one test tube in the triplicate. Okay, take 3 test tubes. Now take a test tube containing a sample. Now in the that test tube you will just take your sample whose concentration you have to determine. In that sample you will add your medium. Okay, that medium, growth factor and all the things you have to add. Okay, now add 2 ml of amino acid assay. 2 ml amino acid assay medium means the bacto amino acid assay medium to all the tubes. Okay, all the 5 tubes. These are 5 tubes. 5 means 5 into 3. Triplicate we will prepare. 5 is a 15 plus 3 here. 3 test sample, it's a 3. That means 18 test tubes. So in that, add the 2 ml of amino acid medium, that is the bacto amino acid medium to all the tubes. Then all tubes are autoclaved. Now go for the autoclave process then, so that all the preparations can sterilize very nicely. Now add appropriate test microorganism. For example, here the Streptococcus facialis 80cc8043 for valine and leucine, Leuconostoc mesenteroides 80cc8042 for methionine to all the tubes. Now after adding the microorganism, incubate 37 degrees Celsius for 17 days. Now set the optical density at 600 micron meter for spectrophotometer and just take the reading for the blank. Okay, and measure the optical density of all test tubes. Okay, now the optical density so obtained are subtracted from the blank. Okay, first you just set your optical density at 600 micron meter. Now after taking after set the optical density at 600 meter, take the optical density of all test tubes with blank also, including blank. ऐसा मैं लिख देती हूँ, including blank. ठीक है? Okay. Now optical density so obtained, whatever optical density just we are finding in the all test tubes, just subtracted from the blank. Okay, whatever optical density we got from the blank, just subtract your density or the reading from the blank and plot it on the y-axis. Okay, so here I will how we will plot? On the y-axis you will just take the optical density, that is the OD, and on the x-axis just standard amino acid concentration, okay. So against the standard amino acid concentration on the x-axis, so the amino acid concentration on the x-axis and the optical density on the y-axis, and just plot your graph, okay. How you will plot the graph? Just keep in your mind, just set your optical density at the 600 micrometer, then just take the reading for the blank and take the reading for all the test tubes, either it is a test sample test tube or the standard preparation, take all the readings, okay, and just subtract these readings from the blank and then just draw the standard curve, okay. Now the concentration of unknown sample is determined by the process of extrapolation of the plot graph. Now optical density is here. Whatever optical density for the test sample we receive, just draw on the y-axis and just extrapolate on the x-axis, and this is the unknown concentration of amino acid, okay. So now you know, now you know that how we calculate the amino acid concentration in the test sample. I hope you enjoyed my lecture. Any doubt, kindly ask me in the comment section. Otherwise, please like, share and subscribe. Thank you.